I really like living and working in Vienna. It's very lively and open city to live in, minus the wind. And yeah, for AIT, I feel like we're very lucky because we have access to a very wide range of different lab equipment that we can use. And also for my supervisors, I have to say they're very, very welcoming and supportive from the beginning and always take time for our discussions, which is great. So yes, I enjoy it very much, especially because we have a lot of different opportunities. For example, now because of pandemic and so on, we have a series of monthly trainings with experts on specific topics, and it's really nice because we can ask them questions directly and also learn from them, which is the best. And also it's really exciting to meet people from different backgrounds with different ideas. So catching cancer in an early stage in order to prevent disease spread through the body is still an unsolved problem in today's clinics, since a lot of times cancer is only detected when pathological symptoms arise. And EVs, or extracellular vesicles, are released by virtually all cells and can be found in easily accessible body fluids such as blood, saliva, even urine. And therefore they could be really valuable for relatively easy detection of the disease. And so because of that, EVs diagnostics from body fluids open a plethora of new possibilities for cancer detection, and our review implications and pitfalls for cancer diagnostics exploiting extracellular vesicles basically gives an overview of the diversity of currently known EV species, cargo, and the relevant cargo molecules representing potential biomarkers to detect, identify, and characterize tumors or tumor cells. So as you know, cancer is a life-threatening pathology where dissemination of tumor cells to other organs across the body significantly metastasis significantly decreases patients' chances for survival. And EVs, or extracellular vesicles, have been shown to have probably also a really important role in this process, where they can, for example, facilitate the cancer spread by establishing so-called pre-metastatic niche. And in order to do any of these things, the EVs have to cross biological barriers. So in our project, what we're trying to understand is the uptake and the processes behind the transport of, in this case, prostate cancer EVs across biological barriers. And for that, we're using different in vitro models. This should help us not only to understand the prostate cancer itself, but in general, cancer EVs and how they can move, and how also biological barriers can react to the EVs. For our biological barrier models in vitro, we are using the Transwell system, and for that we can use all sorts of different cells. Really depends also on the barrier characteristics and the experiment itself. For example, we are using immortalized cell lines such as D3 for the blood-brain barrier, and then, for example, HTB41V2 clone for the salivary gland epithelium. We are using and establishing also by my colleagues the differentiation protocols for the human-induced pluripotent stem cells, differentiating into the blood-brain endothelium. And we also have the possibility to use primary cells, for example, for lymph. So yeah, lots of options. So I always see myself more like in the academia, like also passing my knowledge on and so on, and tutoring others. But let's see, I also don't exclude the possibility of maybe working at a company focusing on 3R field, for example, like organs on chip for the clinical applications. So we'll see.